Kia ora te whānau, no mahara mai ki tāke kōrero I tēnei rā. I'm called Jerusha Bennett tōku ingoa. Ki nā atua nā tipuna me nei iwi o tēnei takiwa, tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora everyone. I just said in Te Reo Māori, the Indigenous language of New Zealand. Hello, thank you for having me, and just an acknowledgement to the Indigenous people of this beautiful place. I am a parasitologist, so I work on worms. I'm very different from most of the expertise of everyone who's talked already. And I focus on parasites of a range of different animals, mainly seabirds and fish. And I'm coincidentally here in Tasmania working with Kate on some of the kanakana projects that we've got going on. So I'm delighted to be here. And I thought the only thing that I work on in Antarctica is squids. So why not share some preliminary data that I've got from a little side project. And this is part of the Emerging Aquatic Disease program that Kate introduced earlier. Now, we all know that the world's ecosystems are changing in response to natural and anthropogenic pressures, hello, climate change. And if we want to know how our systems will change in the future, well, first we need to know what animals live here, how do they interact with each other and their environments. And the way that we look at these systems is also changing. And with growing appreciation of certain underrepresented or previously unknown animals. And in Aotearoa, New Zealand, I'm sure that it's the same here in Australia and also globally. And some people have mentioned it earlier, that we tend to take a reactive approach to disease outbreaks, whereby a disease occurs and there's a race against the clock to try to identify the disease agent, manage its impact on the remaining population, but the damage is already done. The disease has already impacted the hosts. And in our lab at Otago, we take a proactive approach to disease outbreaks by opportunistically sampling host species and inventorying their parasitic fauna before a disease outbreak occurs, so that we're in a better position to assess what parasites are here, what is changing in the future, and what parasites might be most likely to cause problems in the future, and or potentially host switch or be invasive. This is a really common problem that we have in parasitology, disease ecology, is that we don't know if something's new. We don't know if something has recently switched, or if it's been here this whole time impacting a host, and some sort of environmental factor has exacerbated its ability to cause disease. And I'm just going to give you one example of a project that we've been working on in a proactive capacity to try to provide this baseline data of parasite infections in some animals. And this particular project is about arrow squid, Nototodara saloni. It's a really important prey species and predator in ecological systems, and for a range of different predators like sharks and seabirds and marine mammals. And it holds really important ecological roles in any of its environments that it lives in. And for New Zealand and around the world, it's really important economically, and because we like to eat it. I did. I still do. And in New Zealand, it's our third largest fishery in terms of the monetary value that it brings in. So despite the fact that it is really significant economically for us, we know very little about what parasites infect these animals. And the little that is known on squid, I suppose worldwide, is that they can host zoonotic parasites from the genus Anisakis. It's a nematode that can accidentally infect humans. So I'm just going to share some results of what parasites infect Aotearoa's arrow squid. And the samples came from a trawl survey that was contracted to one of our institutes, NIWA, by the government to assess the hoki stocks down in the subantarctics. And in the blue here is just the areas where some of the samples that I'm going to talk about were collected from. And this was a routine trawl survey that occurred in November last year. And of this, I'm going to present some results from 70 squids. And we use DNA and morphology to identify the parasites that those squids had throughout their body in different organs and in their body cavities. And this is a picture of myself and my colleagues. This isn't an arrow squid. The arrow squid only get to about this big, but it was a new species, which was really cool, of squid. And so in terms of parasite diversity, we found 5 different parasite species. The first, this is a larval cestode called Clistobothrium. This parasite can be about 3 centimetres long, and this is a larval stage that infected 82% of the squids. And every squid that was infected with on average, about 10 individual cestodes. Anisakis, this is a nematode that I mentioned earlier, the one that can cause infections in humans. This we found a range of parasite species of Anisakis, but I've grouped them all together here. And 95% of the squids that we looked at were infected, and with a mean intensity of 15 worms per squid. And we found a couple of other minor parasites. Another nematode called Lepetoscaris, and 2 trematodes, one from the family Icoselidae and another one from suborder Hemierata. We are still working on the identifications of these, but I'll talk a little bit about what we can surmise from this shortly. And then, so because these 2 parasites were the most commonly found, we focused on these when we were looking at what can these parasites, looking at these parasites in terms of their infection dynamics. In terms of the squid sex, we found no difference between the abundance of nematodes and cestodes between the sexes of the squid. If we had, this could have suggested to us that potentially one of the sexes has a different behaviour, and that's why the squid are eating different things, and therefore they could have different numbers of parasites. But that wasn't the case in this instance, and we also found no relationship between the size of the squid and the number of parasites that they hosted. But in terms of the site of infection, so where we found the parasites, this is important when we're looking at what part of the parasites people eat, which is the mantle in green, in the body cavity. And we looked at where parasites were found within the different organs. For cestodes, we found big differences in where the abundance of parasites within the different organs that I've shown up here, with the highest number of cestodes being found in the cecum, which is a sub part of the stomach. And we found a similar story for the nematodes. But the only difference between the nematodes and the cestodes is that we found more nematodes in the stomach compared to the cestodes. And you can see that the parasites, nematodes and cestodes, are in all of the organs, and or the digestive organs at least, and they all have the ability to embed or attach to the body cavity, which is the part that we consume. And so just what does this tell us? Well, it's clear that these parasites do warrant further study, and to be able to look at the zoonotic potential of the nematodes, the Anisakids, because we eat them. And yeah, this is just a picture of someone's intestines with a nematode attached. And not only can people be accidentally infected by this parasite, but often it's the case where you may consume a squid or another fish that's also infected by this parasite, and you have an allergic reaction to the cuticle, the outer skin of the parasite as well. And then we have also gained some insights into squid ecology. There we go. So for the cestode species on the right, this parasite I mentioned is in its larval stage, and in this stage it is waiting for the squid to be consumed by the next host, which in this case is the long-fin mako shark. And so this suggests that these squid may be important prey species for that shark. To be infected by a parasite, you have to eat something that's already infected. And so this is how these parasites travel through the food web to complete their life cycles. And it's likely that this squid may be an important prey species for the mako shark. And the same thing with this trematode that we found. The species in this family are known to infect sunfish as their final hosts. And so it's likely that these sunfish are also consuming squid in some capacity for the parasite to mature and reproduce, which is really interesting. But the main point, and I feel like probably the most relevant for us in this room, is that studies like this are really important for us to compare against in the future. And in Aotearoa, New Zealand, of all marine animals that we expect to host parasitic organisms, less than 4% of our animals have ever had any records of parasites. And if we look at the cephalopods, less than 2% of the cephalopod species have records right now. So yeah, this data is really poor. So it's really important for us to create these databases so that we can compare in the future. And I just wanted to note that we have also been looking at the diversity of these anasakids in a range of different deep sea squid species as well, which is really interesting to look at the different haplotypes and which ones specifically are the zoonotic ones, because not all of the species are. Yeah, and I feel like despite what our backgrounds are or what we specialise in, parasites are very specific. And, you know, we all have a common goal, which is to get a better understanding of what's happening now so that we're in a better position to protect or safeguard the future of our wildlife and our connections to that wildlife. So this is just one example of those. An example of the potential of proactive investigations into diseases or parasites. So yeah, kia ora, thank you.